All right, hey guys, I wanted to make a quick video for you showing a pretty complex tool that we're going to be using a couple times. First time, obviously, right here in evolution, but then we're also going to be using this as we go into molecular genetics to do some really interesting things. The resources that I would like you to take a look at today and the two things that I want to show you are these two links. These are links that the public can use, that also people who are doing real research in evolution and molecular biology use pretty consistently. This first link that I would like to direct you at, this is on the evolution page. For you guys building your cladograms today, you're going to need to use the National Center for Biotechnology information, and then you're also going to use this thing called the Basic Local Alignment Search Tool, or BLAST. Not the last time that we'll use this, so we need to learn how. So this is just a tutorial on how to use these two tools. Both are websites. This one will serve, obviously, a different function from this one. So let's show you exactly what you're going to be doing. You're trying to construct cladograms today using some specific genes that you're interested in looking at. So that first problem that I have for you guys, I'm telling you what to type in here, but the second problem I want you to explore a little bit. So what we want to type into the search bar is, this is basically like Google for genes and genes that have been sequenced. So we can look up almost anything we want. And the first thing I would like you to check out here as our first example is just type in human. And then we're looking at this protein called catalase, which breaks down hydrogen peroxide inside of our bodies, because that's a nasty toxic thing. We'll do more with catalase later on in this course as we learn about enzymes. But that's what this thing is an enzyme. So I'm going to type in human catalase, and then I'm going to hit enter, and it's going to search for anything that might be human catalase in any of the databases that they have. So it's going to come back with some results just like Google does, and you can see it has 346 results. It has 346 hits, just like Google would have. And then it's going to say catalase, Homo sapiens, and it even tells us exactly on our chromosome where that gene is located, which is kind of cool. Next, what I'm going to do is I'm going to click right here where it says CAT, this gene identification, and I'm going to hit that link, and then it's going to pop up more specifics about the gene that we're interested in. It gives you the lineage, so we're eukaryotes, and it goes all the way down to our genus and species here. You can see mammalia, and we have primates, exactly where we're at, so that's interesting that they bring that up. And a little summary of what this thing does. Where we're interested in right now is to scroll all the way down through this information. And this will make more sense as we get into molecular genetics, exactly why we're going all the way down here. But we want to go to mRNA and proteins. That's what we're interested in. We're interested in coding for the mRNA and protein and what information there is there. So we've got two lines here. Where I'd like you guys to click is just on this first line right here. And what that's going to bring up is the specific, some of the more specifics with this gene. I want you to click on FASTA, then this next link that's up here, and then this should look familiar. What does that look like? That's the actual code for catalase. So what we're then going to do is select all of this, the entire code that makes up human catalase, and we're going to hit copy, and we're done with this first website. Now where we want to go is over here to the BLAST, using the BLAST website. What we just copied, all that information, was a nucleotide sequence for that specific gene. So we're going to hit nucleotide blast. You can also look at protein blasts, but we're dealing with nucleotides right now. Up in this box, all I'm going to do then is Command V. Boom. There's all of my code for human catalase. I'm going to scroll down and select a few things. Here's the database, so it's choosing what do you want to look for out of all of the information that we could possibly scroll through. Do you want to look for just the human genome? Do you want to look for the mouse genome, which is a commonly used animal, as you guys know, in biological research? Or do you want to look for others also? We want to make sure that we're selecting for others, because we want to look through all sorts of different organisms that have this catalase gene. We want to see how closely related ours are to these other organisms. So we want to make sure that that's selected. You can also select down here, you can optimize for highly similar sequences, so I'm going to leave that selected here. But as you guys are exploring a little bit, see what happens when you go to more dissimilar or even somewhat similar sequences. When you select that, you're going to have more results that way if you select for these. But I'm just going to hit this one first. I've got my information plugged in there. Now I'm going to hit blast. And now what this is doing is this is going to take a little bit of time. It is searching through every possible sequence that's in this database, and it's aligning the sequences to see how similar they are. So as you guys practiced that yesterday using that Howard Hughes Medical Institute sheet, where you had these sequences being aligned with all sorts of different organisms. That's what this is doing right now. It's taking all of that information and going through all of the database, and it's trying to align it to tell you how similar the sequences are between different organisms. So this might take a little bit of time, so give it a second. Be patient with it. Here are our results, right? So we've got all of this sequence. We had 2,300 letters. These are the results that we're getting. And as you scroll down here, this is where things can be a little confusing, but I want to point out the things that you should be looking at. Your max score, the higher your max score is right here, the more closely aligned the sequences are. You can also see how closely aligned they are here based on percentage. So it's no surprise that the first thing that we pop up is we see that in a database is in the human kidney for catalase. Catalase and the mRNA sequence, obviously we have a really high match because, well, that's what we searched for, right? So that's the first things that it pulled up. But then what's interesting is if you scroll down through this max score, it lowers and lowers and lowers. And you start to see these things that are different organisms pop up. For instance, if I want to click on this one, it's going to tell me how closely aligned it is to this genus and species, and I have no idea what that is, so I can just do a quick search for it. So I'm going to highlight that and copy, and then I'm just going to hop over to Google. Right, and let's throw that in real quick to Google and see what is this thing. This is a Sika deer. Okay, never heard of a Sika deer, but this is how closely aligned the human gene for catalase is to Sika deer. We've got a 90% match. And as you scroll up, you can look through all sorts of different organisms, see how closely related our gene is to their genes. And what's really cool too here is if you look through sequences and sequence data, you can see we have a match of your data that you searched, your query of C, matching up with C of this particular subject, and then you can see where the gaps are and where the letters and the mutations have changed, which is changing this percent identical that we have up here. From there, all I want you guys to attempt to do is construct a cladogram using this information. So it's a really, really powerful tool. The purpose really of doing it the first time is to practice building cladograms using DNA information. But this is also great practice for what we're going to be doing later as we go into molecular genetics and you guys start sequencing some of your own data. Okay? If you guys have any questions, I'll be walking around in the room today. Come find me.